coming here, and I'm really sorry that this is the only room we could get at such short notice. And I thought at 3 o'clock on a Friday afternoon on the last day of the quarter that we wouldn't have this many people here. I should have, of course, remembered that we have Ian Wilmut. And we're really honoured, actually, on Einstein's birthday to have another famous scientist who's spanning the 20th and 21st century. I'm expecting you to live a long time with the stem cell work. Ian, of course, is best known for being the individual who cloned Dolly the sheep way back now in 1997. It's 11 years ago. Dolly, of course, herself was born in 1996, but nobody got to know about her until February 97. And Ian has been working pretty much all his life on reproductive physiology. He grew up thinking he was going to be a farmer, loved the outdoors, but really didn't like the management side. I can totally relate to him on that. Many people do not know, well, except I put it on the poster, that he was also the first to actually produce a calf from a frozen embryo. That was Frosty the calf, who appeared before, and the other good name, actually it was after Frosty the cartoon, but in the early 1970s. And in fact, he had his eureka moment in a pub in Dublin. Hard to believe, when talking to a Danish scientist. And unlike lots of people who have wonderful ideas in pubs in Dublin, when they wake up the next day, may not sound like such a good idea, but Ian, when he woke up the next day, thought it still was a good idea and went ahead and proceeded to do the work that eventually led to Dolly the sheep. And there was lots of other lambs in between and after. And in fact, his research on the notion of using cloning to produce therapeutic products in the milk of mammals was one of the reasons that he initially, I think, got into stem cell research, to look at the whole idea of using regenerative medicine to work on methods to be able to address some of the dreadful, debilitating diseases that we now have no really good method of treating or being able to cure. So he decided that cloning sheep was passé and there just wasn't enough excitement in animals, so he thought, let's try another area and go into stem cell research. And rather than spending the next 30 minutes talking, I will let Ian tell you the story about his position at the Scottish Research Centre for Regenerative Medicine. Thanks, Ian.
Thanks very much indeed. And for your warm welcome. It's a great pleasure to be back here. My wife and I were both here a few years ago and enjoyed ourselves very much. I'm sure the same thing's going to happen again this weekend. Before I get into the meat of the talk, there are a number of acknowledgements that I must make. At the present time, as Martina indicated, I'm head of a new Centre for Regenerative Medicine in the University of Edinburgh, supported primarily by the University and the Medical Research Council. But as she indicated, I previously worked with farm animals, and the work was done at the Roslin Institute and funded by a variety of different organisations, mostly British, but the European Union is one of them. And you'll also recognise, of course, the Geron Corporation, who funded our research at over one particular period of time. Just because I said Geron, you don't have to. And in the interest of disclosure, I should tell you that we own some Geron shares. I have no other direct involvement with Geron, but simply on the basis of the fact that I own some of their shares, you may feel that it would influence some of my opinions. The second thing is to say that for most of the talk, these are strategies, ideas which were developed with my colleagues, and these are the current PIs in the Centre for Regenerative Medicine. And it's my privilege often to travel and to talk about the things that we're trying to achieve. But they are the source of a great part of the ideas. I thought what I would do is keep this mostly fairly general, indicating the sort of strategy that we have, the priorities that we're giving for the use of stem cells, and how we might take knowledge gained from research with stem cells through to the clinic. But I will also emphasize, give a little bit of details of one of the pieces of research which is critical to the case that I'm going to make. And also, because you won't have heard of the centre, at the end of the talk I'll describe the priorities that we have for our own research in the centre, in the hope that there may be an opportunity to collaborate with some of you, perhaps tempt some of you to come and work with us. once you know what it is that we're trying to do. So the general sort of scheme that people use when trying to illustrate the ways in which stem cells will be used is to indicate that if you have pluripotent stem cells, usually this means cells derived from the embryo, but we'll come on to that later on in the talk. We know that in principle they have the ability to form all of the different cells of the body. And so you could derive them from an embryo which is immunologically matched, let's say, to the patient, and transplant cells into a person in order to replace those which are either have died or not functioning properly. And this really is the general, it's the most commonly advanced argument for research with stem cells. And we think it's much too simple a case, and it's probably much too naive and optimistic in describing the way in which this area of research will contribute to new therapies. And I want to go into it in quite a lot more detail. So the research that we do is concerned with cells which are recovered from embryos, from the fetus, from adult tissues. We work at all three different stages. We do work with human embryos, human cells, and with cells from model animals, of course, most particularly the mouse, but some of our very basic studies are in Xenopus, for example. So we have a fairly wide remit. And work with them at any stage through, from these particular stem cell populations, through the progenitor stage, through to the particular tissue that we might be interested in. And as you would anticipate, we use these cells to look into the mechanisms which regulate normal development, because this knowledge will be one of the main things to come from stem cell research. Looking at things like the signaling mechanisms from one cell to another, the epigenetic mechanisms which stabilize the genetic functioning of cells in particular states and ensure that daughter cells function essentially in the same way that the initial cell does. Of course, as you gain knowledge of these normal functionings, you can then look at the effect of different diseases on these functionings, both individual cells and the relationships between cells, and that in itself will be very important for medicine. There are many, many diseases, and I'm going to come on to one or two examples later on, where we simply don't understand the molecular mechanisms which bring about the symptoms. And even to get that knowledge will be useful in trying to devise new drugs, new treatments, quite apart from any strategy of cell replacement. Of course, this knowledge is also important if you're thinking of working with, let's say, embryo stem cells and deriving cells of particular lineages from them, of being able to derive those cell lines effectively so that you have a high quality, uniform, consistent cell population which will be available for study or for use in therapy over a period of many years. Same thing applies to the need for research in order to produce systems which are reliable for producing uniform, consistent populations of cells for therapy. Usually, if you look at a paper that says that somebody has produced, let's say, hepatocytes from human embryo stem cells, they're meaning a small proportion of the cells have become hepatocytes. And I suspect that nobody yet can really say that all of the cells of any particular culture system are 100% of the cell type that they're aiming for. And that's clearly going to be important if we're thinking of using cells in therapy, particularly if we're going to use them in therapy, but also if we're going to use them in research. And as I've already pointed out, I think that most of us, when we're giving interviews, newspapers, televisions, radio, and so on, tend then to say that we have a new understanding of how to derive, let's say, neurons, dopamine-producing neurons, for example, and say that they will be used, this will be useful knowledge for the development of therapy for Parkinson's and things like this. But let's look in a little bit more detail at what you would need to know if you were thinking of transplanting cells into a patient. You would need to know what stage of development it was appropriate to transplant into the brain, how many cells, how to transfer them, where precisely to put them. Would there be an issue of avoiding immune rejection if you were thinking of putting things into the brain? There would in many other tissues, but what about the brain? And from a very practical point of view, you'd need to know how to ship very large numbers of cells from perhaps just one laboratory to another one on the same hospital site, but perhaps also from a a biotech company to a hospital many hundreds of miles away. You'd have to have trained staff for not only all of the proceedings in producing the cells, but also for using the cells. So it seems to me that there's a huge amount that we need to know about the cells, a huge amount that we have to have learned about how to produce cell populations and to know how to give appropriate quality control measures, for example, to the regulatory authorities, as well as these practical issues if we are going to think of using cells for transplantation. It's not that I'm suggesting we will never use cells for transplantation, but this is by far the most ambitious use of the cells, and we should be cautious about how we do it and expect this process to take a number of years for most tissues. Some tissues we may be able to use quite quickly, but in most cases it's going to take quite a long time to generate this knowledge. And specifically, it seems to us that we tend to overlook the potential role of cells derived from stem cells in drug discovery, and that it would be important and beneficial to us all if we were to highlight this potential benefit and perhaps focus more of our research on this particular aspect. In doing this, we will also learn many of the things that we need to know for cell transplantation. So you can consider using cells for drug discovery as both beneficial in itself and a stepping stone towards later use of cells for transplantation. So if we go back to this slide, which you've already seen before, this is exactly the same knowledge that you would use for drug discovery or for transplantation. There are two different ways in which you might use cells. I think you can classify it at least into two in which you might use cells in drug discovery. One is in improved toxicology procedures, in assessment of drugs which are being developed for any disease. This is a legal requirement, as I'm sure you all know, that drugs have to be put through appropriate toxicology testings in order to see whether or not there are side effects which are unacceptable. And the question which we should all be asking is, are there ways of using cells to make this process more efficient and therefore cheaper? And so reduce the cost of developing new drugs. This is one potential use of cells in drug discovery. The second is in studying particular disease models and perhaps developing, identifying drugs which are able to prevent the symptoms of disease, to treat diseases. They, of course, these new compounds would also have to be screened in the same way. But I think there's an initial step here which is different from that in a standard toxicology screen. You might use a number of different ways. You might be trying to achieve a number of different ends by producing new drugs to prevent cell death or to mobilize cells for repair. There are quite a number of different variations here, which I think have scarcely begun to be investigated yet. So let's think of these two potential uses, toxicology and studies of particular disease in turn. It is a very well documented fact that some drugs face a very late withdrawal because of unacceptable, unexpected side effects. And there is this often quoted figure of billions of dollars a year which are wasted in a sense because of the fact that compounds are withdrawn relatively late on. And the question that essentially we want to try and ask is it possible to reduce this cost by identifying at least some of the unacceptable compounds earlier on in the process? So that I think it's naive to pretend that you would never have a late withdrawal, but you might have a smaller incidence of it and therefore reduce the cost. I think obviously the pharmaceutical companies have been well aware of this potential benefit and have begun to do a little bit of research themselves, which in the case of some companies they're rather reluctant to talk about. But it may be that there's an opportunity here for academics, for spin-offs from universities to provide a service to the pharmaceutical companies in developing these tests and then applying them. The most important cell types that you need for this sort of toxicology screening are the liver because of the differences in metabolism in the human population, and then the heart and the nervous system because of the fact that they're most important if they are adversely affected by new compounds. And we are beginning to learn more about how to differentiate cells of these lineages from human embryo stem cells. And I'll present you some data in a little while about deriving human hepatocyte-like cells from human embryo stem cells. if you think what you would like if you were trying to set up a toxicology screen using cells, you want clearly genetically identical populations, you want phenotypically uniform populations, and ideally you want them available repeatedly over a period of years so that you would know that an assay that you were doing this year was being done with essentially the same population as had been used a year ago or 2 years ago, so you could make a comparison between different compounds on a common assay system. And this simply isn't possible at the present time because primary cultures are used and they are not, cannot be maintained over this sort of period of time. That's the reason why the pluripotent cells, either from embryo stem cells or from reprogrammed cells, in principle offer a totally new approach to meeting all of these requirements. And of course, one of the things that you would really like is to have a range of different genotypes, including some from people who would prove to be vulnerable to particular medicines and had side effects. Just to bring this impact of this home in a slightly different way, death because of an adverse response to medication, even when it's given, prescribed and taken in a totally appropriate way, it's a very significant cause of death. I think it was 10 years or so ago the 5th most common cause of death. I must admit, when I came across that statistic, it made me pause a little bit about taking medicines just as quickly as I might have done beforehand. Now, it may be that in many cases the victims are older people. Okay, maybe I begin to come into that category. And already taking medication for other reasons. So it may be an unfortunate cross-reaction between different medications, which is involved as well. But that's 10 years or so ago. It was a very thorough analysis of a variety of different publications, the 5th most common cause of death. And those who survived, many of them had long periods of time in hospital recovering from an adverse event. So if you have cells from people who are of a known vulnerable genotype, but also meeting these requirements, they would be extremely useful for toxicology studies. And we're going to come on in the end to the idea of using reprogrammed cells. And this would mean that it would be possible to take skin cells, let's say, from somebody who had an adverse response, perhaps one that wasn't predictable, wasn't understandable, but to be able to produce cells from people like that, which would meet all of these other requirements, would be extremely powerful, it seems to be. So the second application of cells in drug discovery is to study particular diseases for which there isn't a treatment at the present time, inherited diseases. And there are many of them. And what we would want to be able to do is to produce cells which are identical to the patient's cells so that you can study the cause of the disease. And that when you then have a molecular mechanism, something that you can measure in a biochemical assay or observe in a microscopic assay, you would be able to use these cells in high throughput screens to look for new drugs. It would then be necessary to test these in animals before you were able to move on to clinical trials. But again, there is a potential huge advantage in using in cells, particularly if you can have cells which are genetically identical to patients who have inherited a disease. There are a great variety of different inherited human diseases. Just a handful here. Motor neuron disease is one in which I became interested a number of years ago. Parkinson's disease, the other neurodegenerative diseases, in fact. Many cancers, of course, and psychiatric diseases. One of my colleagues in Edinburgh is keen to have cells which he can use to study schizophrenia. I have to admit, I've never asked him how he's going to do that, but he is very keen. So let's look in a little bit more detail at the disease which we call motor neuron disease. Amyotrophic lateral sclerosis, or ALS, is I think a more common name in this country, or after Lou Gehrig, of course, the baseball player. In Scotland, if we were christening it now, we would call it the Jimmy Johnstone disease. Jimmy Johnstone was a famous soccer player, pretty much my age, and he played for Glasgow Celtic about 40 years ago when they were the first British team to win the European Cup. Now, to get the context, this is pretty much equivalent to winning the Super Bowl, okay? And and he was sort of lighter built than I am and very quick on his feet and was called Jinky Johnson because he was a winger. If any of you know anything about soccer, he could take a ball round the defenders and score very well. I had the privilege of meeting him a few months before he died. He was a lovely man. He by then would have been early 60s. The symptoms with this disease are that it's a variety of different, it's a family of different diseases, in fact. There isn't just one. But the most common incidents, the symptoms first appear in mid-50s and take 4 or so years for the person to die. Does anybody in the room have a family member who has this? Can you make a noise just in case we're not seeing you? No, I don't think there are any hands going up. Okay, it's a very unpleasant disease. I mean, what happened to Jinky is that he lost control of movement in his legs and was sort of falling over. It usually starts either in your arms or your legs, and progressively you lose control, of your limbs, and then progressively into the trunk. And you die because you stop breathing. You suffocate. When I met him, he was lying on a high-level bed at about this sort of height, not able to move anything south of here. I think that's horrible. There is no treatment. What it says here gives you a rough impression. There's a little bit of debate about precise numbers, but in roughly 10% of cases, it's clearly a dominant inheritance through a family. And in 20% of these, that's 2% overall, this is known to be associated with mutations in SOD1. There are more than 100 different mutations. Actually, I must correct this slide and bring it up to date. Within the last 2 or 3 weeks, there's a paper come out identifying another gene. But up until then, this statement was true, that there was only the one gene known to be involved. One of these loci, they've now put forward a candidate as being involved. Classic genetic disease to identify these loci and then the genes is extremely difficult. And as I mentioned rather casually, the molecular mechanism isn't known, and there is no effective treatment. There are other people, of course, just to illustrate the range of disease. Stephen Hawking, the British astronomer, probably has one of the other members of the family. And he developed symptoms when he was 20 and is still alive just over 60. So you can see that there are a range of different symptoms depending on exactly what sort of genetic mixture is involved in the particular patient. So this audience could work through this. Many of you will know it anyway, but you could work through the logic of it very easily. The first thing that a molecular biologist would do, given the information I've given you so far, is knock out the gene, thinking that it may be a loss of function. It has no effect, so it's not a loss of function. However, if you put one of the mutated human genes into experimental animals, expressing at a high level by using other regulatory elements, you get the symptoms. And so the suggestion is that it's an effect of a corrupted protein, disturbing cell function in some way. But the ways in which that happens are not understood. And as a non-expert, if you go into one of the ALS meetings, there are intense discussions and debates about how this is happening. So it is still, I'll put the sentence the other way around. There is absolutely no understanding, no agreement as to how this corruption causes the problems. So I mentioned that overexpression of a mutated human gene causes the symptoms in animals. And you can see here, particularly in the lower animal here, the back legs are splayed out because the animal has lost the use of those limbs. And the animals like these can be used to test new drugs. It seems to me that in itself raises significant ethical issues. Because the only way you can test a drug to see whether or not it's delaying the symptoms is by giving it to animals, not all of whom will get the drug, not all of the drugs will work. So specifically imposing this on them as a way of learning about potential new treatments for people. And I personally think it's an acceptable thing to do. But I think we need to think very carefully about it. It does raise ethical issues. In this project, we're collaborating with Chris Shaw, who's a clinician research worker at King's College in London. And Chris tells me that using this sort of approach, it's possible to test a handful, literally, not metaphorically, of new drugs each year for tens of thousands of dollars. And that's the current way of trying to develop new treatments. And hence the question, which he and I have been wondering about for a number of years, can we have cells which have the inherited characteristics of somebody with the disease which can be used in screening? So the objective of the project is not to use those cells in therapy, but to have them in the lab to be able to study them and compare them with cells which instead, not also, have the SOD1 mutation, to try to find a common observation of what's going wrong in these cells, so that it would then be possible to use these in high throughput screens. One of the candidate mechanisms is that these corrupted proteins aggregate and therefore prevent normal function in the cells. If that proves to be a key mechanism, then it would clearly be possible to set up microscopic assays to look for aggregation. I guess I'm making one huge assumption there that if I don't point it out before questions at the end, somebody will point out for me. I indicated to you that in most cases it takes 54 years for the symptoms to develop. And we're clearly not going to sit and watch down a microscope for 54 years waiting for the symptoms to develop. So there have to be ways of provoking these changes in an accelerated way in these cells. But people in the field are confident that this is possible, perhaps by stressing them in some way with different oxygen concentration. But it is an important assumption that we're making in advancing this strategy. So then you would take any candidates which were identified this way and use them in animals. The expectation is that you would be able to put through tens of thousands of compounds in a year for roughly the same sort of amount of money as is spent on animal tests at the present time. I think I've already mentioned that these are a number of the other diseases, but there are many others which could be approached using this sort of method of having cells equivalent to those in the person who has inherited the disease, to be able to contrast them with regular cells to look for the difference and then try and find a way of preventing those differences. So then I think we've identified that there are two different ways in which cells might be useful in drug discovery. One is to improve toxicology assessment of any new compound, and the other is in disease models. Let's look into this in just a little bit more detail. I mentioned earlier on that one of the requirements we would have is that the cells should be of a specific genotype in order to be vulnerable to the disease, if that's the particular use that you have. And very obviously, the simplest way in which you can achieve this is to add and overexpress a gene, or to target it in, to human embryo stem cells, a gene which is causing the symptoms of the disease. So in the case of ALS, it would be an SOD1 mutation. And the particular cleanliness about this approach, neatness, is that you would have the original ES cell population to compare those to which you've had added the gene, to make a very, very accurate comparison as to what is the effect of the mutated gene. A second approach for some diseases, and ALS is one of these in some countries, some IVF labs, is to take embryos being produced as part of IVF, which are found to have the particular genotype that you're interested in. And again, this will be for a single gene disease rather than for the toxicology screening. And I'm sure, again, for this audience, you're very familiar with this approach, which is to recover an embryo in the cleavage stage, to remove one or perhaps two cells from the embryo, and then do a genetic analysis to look to see whether the embryo has the mutation or whether it has a healthy form of the gene. Clearly, the idea for doing this is to transfer those which do not have a mutation, but subject to the permission of the parents, the embryos which are carrying the mutation can be donated for research. And that is indeed happening in a number of labs these days. Not to my knowledge with ALS, but it may be that's happening without having been publicized. This is clearly a very effective way of achieving some of the objectives, but you don't have a direct control. The reason why I became interested in this initially is because nuclear transfer is another way of producing cells of the particular genotype. If you have a person who you believe has inherited ALS and you take some of their cells, The benefit is greatest if the patient does not have a known mutation. But if that's the case, you can't actually be sure that they've inherited the disease. Remember, most cases are spontaneous. So it is a calculation, it's an estimate. But if you have a patient who, as best you can estimate, has inherited the disease, if you take one of their cells and put the nucleus into an enucleated oocyte, produce a blastocyst, stem cells, and then in the case of ALS, the different neuropopulations which might be affected, these will be equivalent to those in the patient during fetal development, and you could begin to look for differences associated with the inherited disease. But by far the most powerful way of looking at this is the technique which was developed by Shinya Yamanaka and his colleagues in the University of Kyoto, in which they directly reprogram cells. And I'm sure that you're aware of this work, because to me it is so sensationally important that it is very important that we begin to think about how to use this new opportunity. What they did, put very simply, is to introduce proteins initially into adult mouse cells, which had been modified so that they would only survive if they changed to acquire the characteristics of stem cells. They got a resistance system in them. And they studied the effect of introducing 24 transcription factors. They found that putting them in individually wasn't able to change any of the cells. Putting in all 24 did. And then they worked down the list 10 to 4, and most recently to 3, so that they've identified a small group of factors which are able to change some of the adult cells of the mouse and more recently of humans, so that they acquire the characteristics of embryo stem cells. And you'll recognize the genes which are along the bottom here. The gene which has been missed out in some of the most recent publications is MYC, which is probably a helpful step because it is a cancer-forming promoting gene, an oncogene. Now, there are many caveats that you have to put, perhaps I should go back to this just for a minute, in relation to the original technique that was used. A number of groups have repeated this in the human. Three groups working separately have come up with essentially the same approach and shown that you can change adult human cells into cells which are very similar indeed to embryo cells. In the mouse, Shinya's later paper, which is I think quoted up at the top here, showed that if these cells were selected in a slightly different way for expression of the level of nanog, then they were able to contribute to chimeras and in some cases into germline chimeras, which is the gold standard assessment for an embryo stem cell, so that they really were becoming equivalent to embryo stem cells. But there are a number of caveats which have to be pointed out at the present time. One is that the way in which they introduced the proteins in these early experiments was by using retroviruses, so that on average in the more recent human papers, they estimated that there were more than 20 viral integrations in each cell. And there is a significant risk that one of these has caused a mutation to the gene. But it's surely going to be possible to improve the technique, to find different ways of introducing the proteins, to use viruses which don't integrate. Episomal vectors, for example, would overcome the risk of part of the effect being due to a mutation. And it would be possible by growing out the cells to dilute out the presence of the vector, so that it wouldn't be present in any that you used in therapy. But it does seem to me that at some time in the future, direct reprogramming will become the reliable source of cells. But also it seems important that we continue to carry out research with human embryo stem cells, because at the present time they are the only pluripotent cells which could be used for therapy. Direct reprogramming cells are not yet anywhere near appropriate for use in therapy. And we are still learning about methods for deriving best quality human embryo stem cells and for maintaining them. If you look at the still recent publications, there is quite a lot of variation from one culture to another, which is not understood. There are a number of different hypotheses. So we're still learning how to handle human embryo stem cells, and the knowledge that we gain from this research will in its own turn be beneficial and essential, in fact, for research with reprogrammed cells. And of course, embryo stem cells really provide the gold standard against which you would want to compare directly reprogrammed cells. So I think anybody who's saying we should abandon research with human embryo stem cell derivation is being a little bit premature. But if you make the leap and assume that at some time in the future, we don't know when, iPS cells, induced pluripotent cells, as these are called, are acceptable for use in therapy, then it would be possible to think of banking cells of known genotype, because you know who they've come from, to be representative of the population. And this would be a way of having cells with minimal immune rejection, perhaps with a not too large a number of cells being required in the bank. Before that, of course, exactly the same benefit of having cells of known genotype means that these cells can be used in research into human diseases and into toxicology. To me, Yamanaka's work is one of the stunning pieces of work of the last 20 years or so. It's really superb, and it was very, very carefully thought out right from the beginning. Now, the other thing that we said a little while back about cells which we're going to need, not only that we should have cells of a known genotype, but that we should be able to produce a particular phenotype reasonably uniformly and repeatedly. And of course, pluripotent cells are the ideal source because of the fact that they have the ability to form all of the different tissues. This is not to totally discount adult stem cells, but it is simply to point out the benefits of using pluripotent cells. And as we mentioned before, in the context of toxicology, these are the three lineages which are most important. And I'm just going to present a little bit of information from my colleagues about recent work in producing hepatocytes. These are the groups that have been involved over a period of years. Initially, whilst they were in the Roslin Institute at the time when John Clark was the leader of the group. These are the clinical group. This is the clinical group in the medical school in the university. And as you'll see, this is worked in association with Jaron, particularly Jane Lebkowski. And part of the data that I'm going to show you was collected by Johnson & Johnson. And there's a company in Dundee, CXR, which carried out some of the earlier work as well. So I'm just going to present you some of the work from this group. It was presented recently by David Hay. And I keep on forgetting to put on his name because he used this slide. It doesn't have his name on it, for which I owe him an apology. It has been submitted. Some of it is published in Stem Cells. Some of it is being submitted to PNAS. So what they've done over a period of years is to look at the derivation of hepatocyte-like cells, monitoring the disappearance of expression of key genes associated with particular developmental stages, nanog, brachyury, glucoid, and then the appearance of genes associated with the liver. And later on, a longer series here, you can see more particularly expression of genes associated with hepatocytes, alpha-fetoprotein and albumin, for example, being an indication that they have some of the characteristics of hepatocytes. The work involves the introduction of one of the Wnt compounds, and because this has not been published, we've carefully blanked out the identification of the particular candidate. And you'll see slight differences visually here in the presence of the cells in the tissues from animals, but more particularly here you see data showing the effect of having treated the cells with one of the Wnts during the differentiation process in comparison to the control group, which did not have Wnt present. The idea, remember, is to use these cells in high throughput assays. So you need to be able to handle them not just in comparatively small samples like this, sorry, I was meaning to point like these, but also in 96 well plates and perhaps even beyond that, to try to go down to having one cell at a time. And the work which was, I mentioned, done by Johnson & Johnson is showing the levels of expression of two of the liver enzymes in comparison to embryo stem cells. It's extremely difficult to know exactly which of the cells you should use as a comparison. The primary cultures which are available at the present time are from, I guess, typically from accident victims and so on. So you don't quite know if they are, at a statistical sense, at a normal level. Clearly, embryo stem cells wouldn't be expected to be expressing these enzymes at a particularly high level. So this is perhaps underestimating as a control, but it is difficult to know what to use. It's not very easy to see in the top slide, but as Dave's indicated, in the top picture, excuse me, as Dave's indicated in his slide, the morphology of these cells was normal, of some of them, and as you can see, some of the P450 enzymes are being expressed at significant levels. So, last of all, what I want to do is to talk to you a little bit about the centre. As I mentioned at the very beginning, in the hope that we may be able to establish some collaborations and perhaps to tempt some of you to come to Edinburgh, not only to visit, but also for sabbaticals. This, of course, is an artist's model. This is Craig Miller Castle here. It's well known in Scottish history. This is a Royal Infirmary, which is about 5 years old now, I think, when the major hospital from Edinburgh was moved out of the city centre, out to the outskirts, and the medical school moved with it, so that the Chancellor's Building, as it's called, was established at the same time as the hospital and the Queen's Medical Research Institute, I guess 2.5 years ago now. And that has the capacity for 600 research workers with a reproductive biology group on the ground floor, inflammatory disease on the middle floor, and cardiovascular science on the top floor. And it provides coordinated services for things like imaging, for focal microscopy and things like that. There is a very collegiate approach to research on this site, so that people in these different buildings collaborate in the use of equipment and animal facilities very freely. None of the rest of the buildings shown here exist. This is, I guess, what you would call a proposal for a biotechnology park, which is something which in this part of the world is taken for granted. In Britain, we've been rather slow to catch on. But that will be the Centre for Regenerative Medicine. And I think it's virtually signed on the dotted line that Alexandria, who of course developed Mission Bay, will be taking the next 2 sites for a principal building on that site, which will provide meeting facilities and restaurant, cafe facilities, things like that. Any of you who've been to meetings in Mission Bay will know that they do a superb job of creating facilities like that. And that 3 of the other buildings on there will belong to the university, and the rest will be for commercial development. The centre's been made by bringing together a number of groups who will be known to some of you. The Institute for Stem Cell Research, which was led by Austin Smith until a couple of years ago now, I guess, before he moved to Cambridge, and has a very distinguished record for its basic science research on mouse embryo stem cells, particularly people like Ian Chambers, Claire Blackman, Josh Brickman are in that part of the centre. But it's now many years since the university decided that it was going to create a centre with a broader remit, and recruited not only myself but a number of other people from Roslin Institute, not because we're going to treat sheep, but because at that time an animal science institute had the biggest number of people working with human embryo stem cells. People like Paul D'Souza, people like Dave Hay, whose data I just showed you, for example. We were all at Roslin Institute until 2 or so years ago. And the university has also recruited a number of clinicians with interest in the use of stem cells. Some of them are already using stem cells. And we have also involved the National Blood Transfusion Service, who, of course, they've been early users of stem cells indirectly. But they are familiar with working at clinical grade, which is a key skill which will be essential for any clinical application of cells for therapy. And so one of the things which is designed into our new building is a facility for producing clinical grade cells, and it will probably be under the management of people brought over from the National Blood Transfusion Service. So this is the history, if you like, but the group came together in a centre just over a year ago. It's a virtual centre, unfortunately, because the building on the site that I've just shown you is still 3 years away, and we are in 2 different buildings. But the aim is really to bring together strands of research which are very often in different groups. The basic research on mechanisms of self-renewal, of differentiation, which we've mentioned several times already in the talk, to produce and maintain stem cells at different stages of development. To use these, as I've emphasized a lot already, in drug screening, but also be ready at an appropriate time to use them in cell replacement. I should be careful not to give too negative an impression of our interest in cell therapy. One of my colleagues has just won a large grant to work on using mesenchymal stem cells in bone repair, with a longer-term objective of using embryo stem cell derivatives. So we do have it in mind, and where it's appropriate, we will pursue cell therapy. But we just think that to pursue that without taking full advantage of using cells for drug screening is to miss a huge opportunity. And of course, there will be an interaction between the different groups, there will be an added value, if you like, of having these different groups, the different projects within the same centre, because we all have the opportunity to learn from one another. Unusually, for a British academic group, we have a particular expectation to commercialise our work. Remember the map that I showed you, that we will be on a site surrounded by buildings, which can be let out to companies. 2/3 of the cost of the building is being met by different branches of Scottish government, whose aim it is to create jobs in Scotland. And so I'm quite clear that I have essentially 3 sets of stakeholders that I'm answerable to: basic scientists, clinicians, and government for job creation. And I wouldn't want to get into a fight about it, but these are all important in the strategy of the centre. It doesn't mean to say that many people will contribute in all 3 areas, but that it's important that we set up systems that see that knowledge produced in one of them can easily be transferred across to the other, so that each of us can take advantage of the work being done by the others. It's awfully easy to say. So just to look at the core facilities that we have. I mentioned that the new building will have facilities for clinical grade cells. We're already working at that standard. Paul de Souza is employed by the university, but has stayed physically at Roslin Institute, where they created a clinical grade facility, and is deriving cells. As the regulations can be applied, he is deriving cells which will meet that expectation. We, of course, have access to the clinics, and several of my senior colleagues are clinicians. Andrew Smith will be known to some of you as the person who devised some of the protocols for introducing genetic change in mice. He had a paper in Cell last year in which they'd shifted huge segments of the human genome into mice using a very sophisticated. One of his priorities now is to work with human embryo stem cells, because I think it's still true to say that we lack the techniques for making precise targeted change in human embryo stem cells, and that is limiting many of our opportunity. We have, of course, the techniques for embryology, nuclear transfer. Just to mention in passing, we've abandoned any idea of carrying out nuclear transfer with human eggs because of, to me, the perceived greater advantages of working on direct reprogramming. But we do have that technology in laboratory animals, and have a project working to develop nuclear transfer in the rat, for example. As is necessary for any group of biologists these days, we have a bioinformatician on the staff. We don't actually do microarray work ourselves, but the facilities are available in Edinburgh. The centre does have fax sorting equipment and is just in the process of upgrading it. So I think we have what you would expect of any modern stem cell organisation, but within one group, and very good research workers in each of these different fields. So what are our aims? What are our priorities in terms of research? Well, key to any progress really is to develop new techniques. And so I've mentioned already, I think, Andrew's work on genetic modification. There is a variety of different uses being made of high throughput screening by modern standards on a pretty modest basis, but we have ambitions to develop that more fully. And I think implicitly I've said that we're working on disease modeling and working at that standard. So those techniques are being developed. In parallel, we have work, again, I've mentioned this, working with stem cells at different stages of development and concentrating as much on the idea of identifying and describing their environment, their niche, as well as the cells themselves. And we chose initially to concentrate on 4 particular tissues because of clinical expertise in these areas. We recruited Charles French Constant from Cambridge a few months ago. He has brought with him a group concerned with multiple sclerosis, another neurodegenerative disease. He is a clinical research worker. Alexander Medvinsky may be known to some of you for very basic work on the development of the hemopoietic lineage, and I've already mentioned our association with the Scottish National Blood Transfusion Service. We've recruited somebody from the Anthony Nolan Trust who has a background in leukemia and genetic contribution to leukemia. A number of the people who were recruited a year or two ago are concerned with liver repair and using cells for that purpose. Again, working with cells from adult tissue as well as with embryo-derived stem cells. Clearly, the work that I presented to you in relation to toxicology will also contribute one day to having stem cells, embryo-derived stem cells, which can be used in liver repair. And I've already mentioned the bone grants, the success there where we have a basic scientist and a clinician working together who have recently got a very large grant to use mesenchymal stem cells. This list was prepared not quite a year ago, and we are adding on cardiovascular science. And if you remember, I mentioned that there is a group in the other building with that priority. We have a small grant with them, which will bring in 2 or 3 posts into the centre. And we are working towards having a group concerned with diabetes. Of course, in its early differentiation, that relates very well indeed to the differentiation of liver cells. I'm very concerned that we don't become too diffuse and have one person working on each lineage. But given that you're working in quite a large university with a large research hospital alongside, you realize that there is an eye pavilion, for example, and they are very interested in being able to grow cells which might be used for corneal repair quite soon, and perhaps in the longer term for retinal repair. So that there is always the temptation to collaborate with these people and to try to help them to learn the basis of stem cell research. So what I hope I've done since I first showed you most of this diagram is go through quite a lot of the biology, our views as to how stem cells can be used in toxicology and drug identification, and our own research aims. It is important to recognize that the regulations that we work under have an impact on our progress. And I think in Britain, on the whole, we are quite fortunate. And although there are great differences within Europe in social attitude to stem cell research, the regulations which are coming through at a European level are comparatively open for use of stem cells. Ultimately, it's a decision which is delegated down to each individual country. So we are free to work with embryo stem cells in Britain, even if in some countries like Germany, for example, the regulations are very, very restrictive. Clearly, that's something which will change as we all begin to work with directly reprogrammed cells, which have nothing to do with an embryo. I think it's also clear that we need investment, something which in this state you have a huge amount of, and a great opportunity. In Britain, that's not quite so much the case, but I think both this state and my country, we have a very supportive attitude within our own societies, for which we should be deeply grateful. And I think we owe it to our communities to express, to describe what it is we're trying to achieve in order to carry that support along with us. So I have no doubt in looking forward that we have a great deal to learn. young people in this audience have a fantastic opportunity for careers if stem cells interest you. There is so much to learn. This is such a new area of biology. But there will be enormous benefits come from it over a period of time. We do need effective regulation, and no doubt that will go on changing. Many of you will realize that I'm English originally, but very happy to advertise that Scotland provides a very good environment. It has a very active life sciences community, both in a business sense and in an academic sense. And that there are opportunities for all sorts of partnerships within the United Kingdom, within Scotland. And what's most exciting is that there is great promise for new treatments in the future. And if you think back, and this is where I'm just going to lapse just once into the thing I criticized us for throughout the lecture. If you think of the diseases which have been mentioned at any point during this talk, ALS, schizophrenia, psychiatric diseases, diabetes, heart attack, cancers. I could almost challenge you to put up your hand if you haven't got a friend or a family member who has got one of those. Because, you know, we all know somebody who's got at least one of those. Most of you are so young that you think you're immortal still. But I've got bad news for you. One of those will get you at some time in the future unless we take these opportunities. And then I think in many cases we will be able to provide the first effective treatment. Thank you very much.
I guess I'll ask the first question. You talked about Yamanaka's work quite a bit, but didn't mention Jamie Thompson. He had 4, 2 of which were similar to the genes that they use for the directory programming. Why are you not as impressed?
Yes, I was, but I think that Shinya set about doing this and was the first to do it, and he had a very carefully worked out strategy to make it work. It's not very well publicized, but lots of other groups have clearly been thinking about this and made either no progress or much less progress. So I think he deserves
the recognition.
Yeah, absolutely. I didn't make it explicitly clear, I don't think, that we don't understand how the system works. And some of the genes that are changed, like MYC, like Lin28 and so on, people have a general hypothesis that says that their effect is simply to promote cell division, to open up chromatin and so on, so that the specific transcription factors can then act. But it's just hand-waving. We really don't know how it works, though.
So it's a long time before you really could see that direct programming being an alternative to human embryonic stem cells. So, was there a question? Yeah, sorry.
Kind of sideways from what you're talking about, so I'm sorry if you don't want to answer, but it's curious. I was wondering if you were going from a stem cell to a germline cell.
Yes. Well, certainly, yes. There are a number of groups. It's not something I'm familiar with, but people have produced cells with some of the characteristics of oocytes and sperm by culturing. So yes, that is.
Fascinating question, because then you can get genetic recombination right in the dish.
Yep. It's what, in agricultural terms, Michel Georges called velogenetics 10 years ago. Exactly this, yeah. That you, in principle, actually it occurred to me in a different context. Do we know that meiosis would take place in a normal way in that situation? So we're making an assumption, aren't we? But yeah. In principle, you don't actually have to have the next generation walking around the fields. You could just have their gametes in a dish, 2 dishes, and then mix them and get an embryo, and then get stem cells, and then get gametes. That's what Michel Georges pointed out 10 years or so ago. Question.
Talking about livestock, not humans.
Yeah.
I had a question about feeder layers.
Yep.
One of the problems you know with HES cells, at least getting through GMP and getting to the clinic, has been contamination with feeder layers. What's the status with IPS and feeder layers? Have they been developed so they don't need feeder layers? Have they got defined conditions?
No, I think that as far as I remember, they do use feeder layers. So it's a similar situation. I think we should expect this, though, all sorts of reasons. One is it's at an early stage in development of the technique, and the other is, I didn't mention the number, but it's 1 in 1,000 or 10,000 cells are changed. So essentially what you start out with is, a handful of cells in a dish with others which are going to die. And so that's a pretty inhospitable culture environment, really. Now, how we get around that, I don't know. One possibility, obviously, would be to increase the efficiency, in which case then you'd have more of them in the dish. But I think at present feeders are used. Yeah.
I think it's excellent that you touched on the social aspects of dealing with stem cell research. Like you mentioned, the UK and California are very conducive environments for developing in that area. But as we go into looking at some of the preclinical work and whatnot, and then eventually into using cell therapies as something that we can use similar to drug therapies in the clinics, we are kind of making some assumptions that it's going to follow the same process, but we're kind of in new territory here as far as dealing with a cell situation as opposed to a pharmaceutical product. So do you have any kind of advice, or not advice, but I mean some comments to that regard? Because especially as someone who's so young in the field, and the field is still such early days yet, that's something that we're kind of taking for granted on the scientific aspect, that from a regulatory perspective, that's going to be quite challenging, and yet there's going to be quite the social, and, from the investor standpoint, pressure to get things motivated.
Yeah, absolutely. To be honest, it's not an area I'm very familiar with. The advice that we've been given in the context of any kind of application, whether it's for toxicology, which is our main focus, or for therapy, is involve the regulators early on. Talk to the regulators as soon as you can, because they may not know the answer either, and you may be able to some extent to work with them in order to build up the regulations and help them, to put forward things which are more practicable. But I think something I have thought about rather more is that there are also ethical issues and practical issues. People who have unpleasant degenerative diseases, how to phrase this exactly, probably know that if the system goes through all of its testing, they will have died before the testing has been completed. And so they would willingly take the risk. I can tell you this, I'm not a clinician, just to repeat that, but on the basis of a number of contacts with individuals in that situation, and you can soon see why. It's not entirely, but no, that's dismissive, that's wrong, sorry. It is absolutely not just because of self-interest. They would happily do this knowing that even if it doesn't help them, it would help the next generation of people who have the condition. And they would consciously, do that. But managing that is really, really difficult. Because, what happens, I guess it happens here, it certainly happens in Britain, is at the opposite end of the spectrum, there will be charlatans who will be selling things which, probably have no chance of having an effect. And how a person who has one of these conditions can distinguish between the two is just not at all clear to me. So you're quite right, there are lots of different challenges once we get into the therapy. Now, I guess the same thing will apply here as happens in Britain, but I believe that a similar argument would apply in relation to cancer. I believe that there are different rules, faster throughput situations, as it were, for cancer drugs than for ordinary medications for the same reason. So it seems to me you need to create some sort of alley like that for faster use of cells. But it will be very difficult.
To the pharmaceutical companies themselves to do it too.
Yes. There were many people who think it won't be the pharmaceutical companies, but yeah.
Also just question related to cloning and comparing with IPS, and when all of us biologists, they were told before a decade ago, cloning that terminally differentiated somatic cell cannot be reversed to pluripotent state, and just even though after that new invention and then cloning inefficient continue to be as it is ought to progress. But what do you think with IPS or the direct reprogramming will be the same challenging problem can be facing in the near future?
Yep. I mean, I think you're right to point out the relationship between the two, because I understand that Yamanaka began to think about this direct reprogramming because of the cloning result, along with other people, because the birth of Dolly showed that you can reverse that process in some cells. There are certainly some labs who are still carrying out human nuclear transfer. The practical issue that I have with it is, if you think of the one paper which has been published of ES cell lines from a cloned primate embryo, I think it should be described as one normal cell line from 300 attempts. There was a second, but it was partially aneuploid. And so I think, to be cautious, you should call it 1 out of 300. Now, nuclear transfer is never physically 100% efficient. You always have some sort of loss. But that's probably requesting 30 women to donate oocytes with the hope of having one cell line. And I personally do not think that I can justify asking them to go through that discomfort and very, very, very slight risk. I wouldn't wish to stop other people doing it, but I personally don't wish to do it. If the efficiency went up and became, as it was in the mouse, which is, you'd be close to one oocyte recovery would give you a cell line, close to that. Then that argument would change. As far as iPS cells are concerned, I've already, as it were, nailed my flag to that mass fairly clearly. I have a lot of confidence that it will be possible to make it work, but I may be wrong. There's an awful lot we don't know about the cells yet. For example, about the mitochondria. And there are a lot of things that we need to look at in order to be sure. And so what precisely what we're doing is that we have two groups who will be working with one with George Daly's system, which is the other one which neither of us have mentioned yet, to reprogram. We're setting up a collaborative project with him to study ALS in the way that I did originally with Chris Shaw. So the three labs are working together for that. And I have a colleague who's put together his own system, which is slightly different again, and shown it's effective in the mouse. And we will be using both of these in human cells and studying them in a research context, because that's the way that we will learn whether or not they are normal. It is early to say. You cannot say, but I just hope that will prove to be possible. It's just so much more convenient. I don't think people have worked this through yet. I mean, essentially, if all you're going to do is shake magic dust onto a dish and wait 3 weeks, and you've got the equivalent of embryo stem cell lines, the limit won't be in deriving lines. It will be doing things with them, because it will just be so easy to get cell lines. So it will then, and you will know in advance what their genotype is going to be, which you do not with embryo stem cell lines, with very, very rare exceptions where you've done a PGD. But and so to be able to have lines with a known disease risk, to have lines with a known response to medications, for example, to have a variety of different genotypes which you could bank. I don't know how it works in this country, but you must have a system for bone marrow recovery for donors. People like that can give you an estimate of the sort of number of cells you would need in a bank. And we should be beginning to think about the practicalities of using that approach if it becomes practicable in a few years' time, because I think it would just transform our approach to the use of pluripotent cells. I have never believed in patient-specific cells. If you think that you were going to be, as Wang suggested, producing cell lines from cloned embryos to be the equivalent of each patient, it was just never going to be practicable. The workload is so great. The same thing probably applies for reprogramming. You may want to do that for specific cases, like an inherited disease. If you think of something like CF, for example, cystic fibrosis, if you could find a way of reprogramming cells, getting pluripotent cells from a child with cystic fibrosis, either correcting the mutation or putting in a functioning gene, and then putting derivatives of that into particular critical tissues like the lung, excuse me, sorry. Part of my brain's still on holiday. Sorry. You know, if you could put the cells into particularly critical tissues like the lung, that might be worthwhile. Rudy Yanish and somebody whose name I've forgotten did a proof of principle experiment for sickle cell anemia, didn't they? Which is the same thing in the mouse. So I think in some cases patient-specific cells would be justified and practicable, but in the main it will not. And it will be a question of having a bank which will be a reasonably close match to each of us, possibly with a little bit of immunosuppression. And the iPS cells are just such an ideal way to get those cell populations if the procedure can be worked up to an appropriate standard.
How do you get past the retroviral vector?
Well, an episomal vector or proteins directly or small molecules. Yeah. Definitely soluble. There'll be significant progress towards that goal published within this calendar year. It's just going to happen. Yeah.
What's the starting salary for
You'll be paid in euros, remember, not dollars. Oh no, it's pounds still there. Sorry.
It says, yes.
Forgot.
I'll talk to you later.
Okay, thanks. So I have a presentation here for Ian in honour of his visit to Davis, and I was amused to see you are listed among the most famous Scotsmen. You're right after William Wallace of Braveheart.
That's alphabetical order, is it?
Yeah, it is alphabetical order. That's why. I hope it's not with respect to intent of exactly the destruction he intends to wrought.
I think
Scotland.
I'm lacking in physique as well. Yeah.
So you're hiding behind. Judy wants a picture.
Okay.
So there you go. Thanks
Thank you.
for your
Pleasure. Thanks.
So a few students wanted to.
Yep. Okay.
And that's very good.
Good.
Appreciate it.